Hello dear students, welcome to Practically. We bring learning alive, and in today's session we are going to see 5 most important questions from the chapter Biotechnology and its application. We had already discussed 5 important questions from the other chapters for term 2, so please do check out the playlist. And for now, let's begin with today's session and see these important questions. Beginning with the first question. Name the technique based on principle of antigen-antibody interaction used in detection of HIV virus and explain. So yeah, let's see the technique. Let's see how to write the answer. What all points should it cover? So we all know that HIV is detected by the technique ELISA, which the full form of ELISA is enzyme linked immunosorbent assay. It's given over here, enzyme linked immunosorbent assay. So that is used for diagnosis of AIDS or the HIV virus based on the antigen antibody interaction. And how the ELISA works, where it detects and measures antibodies, hormones, peptides and other proteins present in the blood. Okay, and it works on the principle that when specific antibodies bind to target antigen and detect the presence and quantity of antigens binding. Okay, so this is how it works, and in order to increase the sensitivity of and the precision of the assay, the plate must be coated with antibodies. So as you can see, there is an antigen which is detected by the primary antibody, and to attach to this primary antibody we have one secondary antibody conjugate, and it has a special detector which converts whatever a substrate which we use, chromogenic agent it is called, into the colored one. So if it binds, then this works and you can see the color change which is detected. That is how ELISA works. And yeah, this is the procedure. So first the antibody is attached to a polystyrene plate, which is basically the surface. As you can see, the antibody is attached to that. Then coated antigen is filled with this antigen antibody mixture. As you can see, this is the target protein, and a second antibody specific to primary antibody is then added to it, and then extra things are removed by washing, and then finally the substrate is added which can convert the enzyme to form a colored product which can be measured by spectrophotometry. Spectrophotometry is a special technique which is used to measure the intensity, the color of a particular substance. So these are the steps of ELISA. Moving on to the second question. The development of a transgenic food crop may help in solving the problem of night blindness in developing countries. Name and explain about it. Yeah, so since this is application of biotechnology, so here is an application based question. Let's see. Yeah, so we know what are genetically engineered crops like GMO, though in this the DNA which has been engineered using techniques of genetic engineering. Those are the genetically modified crops. And so we know that night blindness is caused by lack of vitamin A. Vitamin A deficiency causes night blindness. So a special rice has been developed by, which is also called as golden rice, okay, which is a genetically engineered rice which has beta carotene, which is a precursor of vitamin A. Okay, so beta carotene can help make vitamin A. So as you can see, this rice, you can see it's also yellowish in color because of the presence of high levels of beta carotene, which imparts this color. So this is basically a rice with high content in vitamin A, and they are yellow because of beta carotene, and also called as golden rice. So this can help to cure the problem of night blindness. So this is one of the application of biotechnology. Next question. Explain how Eli Lilly, which is an American company, produced insulin by recombinant DNA technology. Let's see. Yeah, so we know that the original insulin, it contains chain A and chain B, which are connected by disulfide bridges, right? As you can see here. Okay, it's not given here, but basically there is chain A, there is chain B. They are connected by disulfide bonds, and the pro insulin has one more extra stretch of C polypeptide, which is connected to them. So it has to be removed, then only A and B will make the original insulin. So insulin was earlier synthesized as prohormone, which has an extra stretch of C peptide. Okay, so C peptide is absent in the mature insulin. So Eli Lilly took an approach in which they prepared A polypeptide separately and B separately. So that's what they prepared two DNA sequences corresponding to A and B chains of human insulin, and then these two DNA sequences were then connected them together. Okay, and they use the normal recombinant DNA technology to do this process. So that is what we have to explain. And what is the main difference in two insulin? It is that which is synthesized as prohormone containing C peptide, which is removed in the mature hormones, whereas insulin synthesized by the RNA technology do not contain C peptide. That is the advantage. This is a process. Okay, they will take the human cell, they will isolate DNA. From the DNA, they will isolate the insulin producing gene. They will take plasmid from bacterium, they will cut both of them with same restriction enzyme, and then they will join together by using ligase enzyme. Then they are going to put this plasmid into the recombinant plasmid into the bacterial cell, which is going to grow and produce that. We are going to grow it in the bioreactor, and then we are going to obtain insulin, which we can inject to the humans. Next question, why is the introduction of genetic engineering lymphocyte into ADA deficiency patient not a permanent cure? Suggests a possible permanent cure. Okay, so we all know that ADA is adenosine deaminase deficiency. So that is a disease which is hereditary, genetic rather. So how this can be cured? Okay, so genetically engineered lymphocyte are added is not a permanent cure, because lymphocytes are not immortal. Every cell in a body has a lifespan. Okay, so just like RBCs have a lifespan of 120 days, similarly other cells also have some lifespan, and after that they die. So if we introduce the genetically engineered lymphocytes, after some time they are going to die. So we need then it cannot produce that particular protein which is required to cure ADA. So so gene therapy was normally used, which cures this disease of severe combined immunodeficiency, that is also called as SCID. So in this, the normal ADA gene copies are produced by cloning and then replaced by ADA. So here is the entire flow chart, as you can see over here. Here we have the bacterium carrying plasmid with cloned normal human ADA gene. Then we have the genetically disabled retrovirus. We are going to add this, clone them together, and then we are going to add this into this retrovirus infects the T cells, okay, the lymphocytes, which is going to put the ADA, the gene which is required for it, and then we culture that, and then we inject that into the child which is having does not produce ADA. So this is how it is done. And what is the permanent cure for that? So if the gene isolated from bone marrow cells producing ADA is introduced into embryonic cells at early stages, it could provide a permanent cure. So that has to be done at very early stages, that too from bone marrow into embryonic stages. Okay, that can be a permanent cure. Here we have next question, next important question. How can gene expression be controlled with the help of RNA? Yeah, so this is an indirect question. You'll have to think directly. You might not find this kind of question asked, so that's why it's important. In this, basically we have to write about RNA interference, RNAi. Okay, so we have already seen in maladie incognitia this technique is used. So this involves gene silencing of specific mRNA, which is done by binding complementary double-stranded RNA to mRNA, which inhibits the translation process, and by that it also inhibits the viral antigen gene expression. Okay. For example, the agrobacterium vector forms complementary double-stranded RNA. So if it forms complementary double-stranded RNA, then it cannot undergo translation. It will not produce protein. It will stop. The mRNA cannot get processed further. That's why it is called as silencing. Okay. So that is how the gene expression can be controlled with the help of RNA using RNA interference technique. So yeah, that is what the mechanism of RNA interference given. It depends upon the marks of the question. If the question is of 2 marks or 3 marks, you can just write it in short what points are there, or else you have to answer that in detail. So yeah, here are some points, important topics from this chapter which you shouldn't miss. First is genetically modified organisms. RNAi is very important, as we had seen one question based on it. Genetically engineered insulin. Lot of questions are asked based on insulin. The diagram of insulin, pro-insulin and insulin, both you need to know. Gene therapy, ADA. Again, the flow chart, please do practice. If you can at least draw flow chart, it will help you to express your answer. And then ethical issues, which is based on biopiracy, that part. Okay, so make sure you prepare well from these topics, and if you have any doubt, any query, do leave a comment and we will be there to help you out. So thank you for watching and stay tuned with us. We are having 2 more chapters of which we are going to discuss important questions. So do stay tuned with us and see you all in the next session. Thank you.